What's going on, everyone? Today we will be discussing cellular transportation. So we're going to be talking all about how ions and molecules move in and out of our cells, as well as how things are transported across the cell. Before we discuss things moving in and out of the cell, we have to discuss permeability, which is the ability for a molecule or an ion to cross the plasma membrane. And our membranes for our cells are selectively permeable, meaning that some things can move freely across the membrane and other molecules and materials are impermeable, or meaning that they are not able to cross the plasma membrane. So you're going to see that some molecules, some ions are going to be able to be transported from one side of the plasma membrane to the other, but they may require a protein or a transporter to help them get there. Other materials, such as water, are going to be able to move in and out of the cell without the help of a transporter. So today we'll be talking about all the methods and all the ways these molecules and materials move in and out of the cell. So here we see what materials are able to move in and out of the cell. And so you're going to see that nonpolar molecules such as O2, CO2, lipid-based molecules like steroids, they're going to be able to move freely across the plasma membrane. And why is this? It is because that, remember, these fatty acid tails that make up the lipid bilayer are also nonpolar. So nonpolar molecules are able to cross freely in and out of the cell. Also, small uncharged polar molecules like water and urea are also able to cross the cell, while ions that contain a charge, such as sodium, potassium chloride, and molecules like glucose, which are uncharged polar molecules, they're not able to cross the plasma membrane. Why is it that a small uncharged polar molecule can get across, but ions and large uncharged polar molecules can't? Well, first, you can determine that the size of the molecule is going to matter when it comes to diffusion. So while smaller uncharged polar molecules can get through the plasma membrane, large uncharged polar molecules such as glucose are going to need a transporter to get across because they are not able to diffuse. And so the difference, though, between small uncharged polar molecules and ions, again, is the charge. When you think about water, a small uncharged polar molecule, it is just having these partial positive and partial negative charges due to the polar covalent bond. The whole molecule itself is not coming with less or more electrons than normal, so it doesn't have an extra charge. So these small uncharged polar molecules are able to actually permeate the plasma membrane. However, ions are formed from ionic bonds, and so when these bonds cleave apart, it leaves these atoms with either more or less electrons, giving it a positive or negative charge. And these charges prohibit these ions from moving past the nonpolar lipid bilayer. The movement of ions and molecules are based on a couple of things, and one of those is what's called a concentration gradient, which is the difference in concentration of a chemical from one side of a membrane to the other side of the membrane. And so when we look at the inside of our cells and the outside of the cells, there are different concentrations of molecules and ions, which creates a difference, a gradient between one side and the other. If we see in this picture here, the membrane is closed, so you're not going to have a movement of ions. But I want you to remember, if we are able to open up these membranes and allow ions and molecules to move back and forth, we are going to see that these charged ions are going to move down their concentration gradient, meaning that they're going to go from a place of high concentration to a place of low concentration. And, these are really small elements, and you may be like, why is this so important, the movement of molecules from one side of the cell to the other? Well, it turns out that everything from the communication between your neurons, the ability for your skeletal muscles or your heart muscles to contract are going to be based upon the movement of these little tiny ions. Not only does the concentration dictate the movement of these ions, but also, since that they come with a charge, there's going to be what's called an electrical gradient as well. And so as an ion moves in and out of the cell, so does its charge. And that creates a difference between charges of the inside of the cell and the outside of the cell. We just discussed that ions are going to move based on their concentration, but they're also going to move based on their charge. And so what we're going to see is that if we have a positive ion on the extracellular space and a door opens up, then that ion is going to want to move to the negative space and vice versa. And what we are going to see within our cells is that the inside border of the plasma membrane is going to be lined up with negative charges. So the charge on the inside of the cell is negative, while on the outside of the cell, on the extracellular side, what we're going to see is on the border of the plasma membrane, all these positive ions on these positive charges are going to mirror the negative charges on the inside. And so not only is the movement of these ions based on its composition, but it's also going to be based on the electrical gradient as well. And so that makes what's called the electrical chemical gradient. And so the movement of ions is based not only on electrical properties, but chemical as well. And so how does this work? Let's say we had a membrane, and on one side of the membrane, and I'm pulling up a little bit different example than what's showed here, but let's say on one side of the membrane we had 100 ions of sodium, right? And right now on the inside of this membrane we had 0. So what's going to happen if you just took into account the concentration is that, this would open, some sort of door in the plasma membrane would open, and these ions would start moving from a more positive and highly concentrated area to a negative and lower concentrated area. And if this basically door or portal remained open, you would reach what's called equilibrium. And so you would probably have roughly, if there were just sodiums here, roughly 50 on this side and 50 on the inside at any given moment. But you have to keep in mind that there are other ions inside and outside of the cell. And what comes with those ions? More charges. And so when a door opens and an ion is able to move in across the cell, not only is it going to be based on the concentration of inside versus outside of the cell, but the actual electrical charges by all of the ions inside and outside the cell. So materials are going to move in and out of the membrane, and either they can move downhill on their electrochemical gradient, which is called passive transport. So in the example used earlier, if we had a sodium ion and a channel opened up on the plasma membrane, that sodium ion, which is in a higher concentration in a positively charged area, is going to move into the cell with a lower concentration of sodium ions and a negative charge. That doesn't require any excess energy being put in. That ion will just move downhill on its gradient. However, if you're trying to move an ion uphill on its gradient, that is called active transport and is going to require energy being put in order to do so. And typically, what will happen is that a protein will use ATP and use that energy to then transport the ion uphill. So what you are going to see when you randomly mix particles together is something called diffusion. And so that basically means that it is the movement of all these particles due to their gradients. But eventually what's going to happen is that once these ions and all these particles start moving, is that you're going to reach somewhat of an equilibrium based on chemical composition and chemical charge. And this diffusion can be impacted by many factors, including the steepness of the concentration gradient, the temperature, which increases diffusion, the mass of diffusing particles, so larger particles are less likely to be diffused into a solution than smaller ones. Surface area is also going to matter as well. The more solvent you have, the more likely then particles can then diffuse. And then also diffusion distance, or the barrier. So if we think about our plasma membrane, if the skinnier, the less thick that the barrier is going to be, the easier it's going to be for ions and particles to move. The rate of diffusion is then calculated by something called Fick's law. And this is something that I'm not going to have you really memorize, but just know that the flow or movement of particles is going to be based on the surface area, so how much solvent you have, the size of the particles, the concentration gradient, or the changes in partial pressure between one side and another side of a barrier, and then also the thickness of the membrane or barrier for diffusion. So for particles or molecules moving down a downhill gradient, there are three different types of passive diffusion: simple diffusion, facilitated diffusion, and osmosis. First, we have what's called simple diffusion. And simple diffusion is going to be when a particle or a molecule is able to move freely from one side of the plasma membrane to the other side of the plasma membrane without the help of a transport protein. And so non-polar molecules and small uncharged molecules will be able to move from one side to the other. But other particles, ions, and molecules will require a protein, and that's called facilitated diffusion. And so a channel-mediated diffusion is going to be when a solute moves downhill on its concentration or electrochemical gradient across the lipid bilayer through the use of a membrane channel. And typically these channels can open and close based on charges. And so, if it's in the open state, this ion is going to be able to move down the electrochemical gradient. But sometimes this channel is also closed, and that's going to then prohibit these ions from moving. Last, we have what's called carrier-mediated diffusion. And the difference is going to be that instead of the ion or the molecule moving through an open pore, the molecule or ion is actually going to need to bind to this carrier protein. And then once the particles bind to that protein, there's a conformational change which allows it to come inside. And so it's not an open pore. There has to be this binding and then change in the protein shape in order to allow these ions and these particles in and out of the cell. So when you're using a carrier to transport molecules from one side of the plasma membrane to the other, there are going to be certain properties that impact how those molecules move. And these are going to be very familiar because these are properties of a lot of enzymes and proteins in general. Remember that specificity plays a huge role in part with ligands binding to proteins. This is the solute that needs to be transported from one side of the membrane to the other. It's only going to bind to that carrier-mediated protein that it fits to. Saturation is going to impact movement because the bigger concentrating gradient you have, the more concentration on solutes on one side compared to the other, the faster those solutes are going to move. You also have what's called competition. Let's say two solutes can bind to the same place. It's going to then depend on whether or not there's a concentration of other competitors that dictates how fast the original solute is going to move. Osmosis is going to be the movement of a solvent, in our case water, through a selective permeable membrane, and this is going to happen due to water channels that exist inside the membrane and because of simple diffusion. Water is going to be able to move across a selective permeable membrane, and once we start adding solutes to a solution where there is a membrane, the water is going to want to move to the solvent is going to want to move to where these solutes are in higher concentration. And that movement is due to hydrostatic pressure, or the pressure exerted by the liquid. And now the osmotic pressure is going to be the minimum amount of pressure needed to stop the movement of water to that highly concentrated area. We characterize the amount of dissolved particles within a solution as osmolarity. So you can use the amount of particles in a solution to determine if solutions are hyperosmotic compared to one another, hypoosmotic compared to one another, or isoosmotic compared to one another. And so here we see that in solution X, we have a greater concentration of these particles within our solution, making it hyperosmotic to both Y and Z, while Y and Z are hypoosmotic to solution X. But since solution Y and solution Z have the same osmolarity, they would be considered isoosmotic. We just discussed all the ways that particles, molecules, water are able to move across the plasma membrane on their downhill gradient. But sometimes there is a need to move particles and molecules on their uphill gradient. And so in order to do that, you are going to need to spend some energy in order for that to occur. And so that is called active transport. And in order to move those molecules uphill, either we're going to use primary active transport, which is when a protein is going to use ATP as its source of energy, or secondary active transport, which is where we're going to use the electrochemical gradient of another solute in order to move a different solute. During primary active transport, a carrier protein, also known as a pump, is going to hydrolyze ATP in order to transport a solute uphill. And so here down below, we see one of the more prevalent pumps called sodium-potassium ATPase, or the sodium-potassium pump, which cleaves ATP in order to move ions uphill. And so here we see that sodium is going to be pumped from the lower concentration intracellular space to the higher concentration extracellular space. In order to do that, ATP is hydrolyzed and that energy is used to move sodium uphill, and now potassium can bind to this carrier protein, and then once this phosphate group is cleaved off, the conformation changes again and allows potassium to go into the intracellular space. So pumps are just going to use ATP as a source of energy to move solutes uphill. Secondary active transport is when solutes are going to hitch a ride based off the electrochemical gradient of another solute. And so in the example here, we see that sodium is binding to a carrier protein, and then another solute is going to bind to that same carrier protein. Now, sodium is moving downhill on its gradient, and this solute is using sodium's electrochemical gradient in order to move uphill. And so we have these two separate methods of getting solutes uphill, either by using ATP or by using the electrochemical gradient of another solute. We have different types of secondary active transport, and the name of these proteins are actually going to change based on the direction of ions or the direction of molecules. And so when the solute or the molecule is moving in the same direction as the solute we're hitching a ride from, if they're moving both from the extracellular space to the intracellular space, that protein is going to be called a symporter. However, if your ion or your molecule that you're trying to move is moving in a separate direction than the solute you are taking advantage of, that's going to be called an antiporter. And so these proteins are named either symporters or antiporters to say it is moving in the same direction as the solute that we're using, or we're moving in opposite directions as the other solute. Many materials are going to need to be transferred in and out of the cell by way of vesicles. And vesicles are formed by using membranes in order to encapsulate certain solutes and materials and then transport them in, out, or across the cell. There are two types of vesicular transport across the plasma membrane: endocytosis, where we're bringing outside materials into the cell using a vesicle, or exocytosis, where we're bringing materials from inside of the cell and dumping them into the extracellular space. Receptor-mediated endocytosis is going to be when a ligand on the outside of the cell binds to a receptor on the plasma membrane, which then triggers a vesicle to form, and then that vesicle can have certain routes. And so typically it's going to fuse with an endosome, where there could be recycling of those receptors enters to the plasma membrane, or it can diffuse with something called a lysosome, in which that the materials inside are going to be degraded by certain proteins. Phagocytosis is going to be when the cell is able to engulf large particles such as bacteria or microbes and basically dispose of it. And so here you see that this microbe is bound to a receptor on the cell, and that triggered the cell to form a vesicle known as a phagosome. And then a lysosome can then come along and fuse with this phagosome, and the digestive enzymes on the inside are going to be able to break apart and destroy this microbe. And thus basically the cell is able to get rid of threats and large particles by doing so. Bulk phase endocytosis, also known as penocytosis, is when there is a vesicle formed around extracellular fluid, which is then engulfed by a vesicle, and then a lysosome can then come along and then break apart, and then the cell can digest those solutes. So endocytosis is bringing in materials into the cell. However, cells also need to transport things out of the cell into the extracellular space and into the bloodstream. So in order to do that, vesicles can also bind to the plasma membrane and excrete certain materials known as exocytosis. And so the vesicles that are formed around these material that are going to be exported out, these vesicles contain proteins called snares, and these snare proteins are responsible for tethering vesicles to the plasma membrane and then allowing the fusion of this vesicle to the plasma membrane to allow for these particles to then leave the cell and enter the extracellular space. If you think about sweating, that materials inside of your bloodstream, that blood plasma and the electrolytes within your blood are transported across the skin, and so we call that transepithelial transport, where movement of solutes occur across epithelial cells. And so in that example, you are secreting out a certain amount of plasma and ions, and so that comes out as a form of sweat. And certain materials can also travel across an epithelial cell from a lumen, let's say inside of the body, to the bloodstream as well. And so that just transportation across epithelial tissue, that's called transepithelial transport. So it's important to know these two terms because there's going to be many materials that move from cells to the bloodstream and then from the bloodstream to the cells. Absorption is going to be when a solute moves from the lumen of an organ into the bloodstream, right? So think about when we digest food and liquids, all those materials get broken down in the stomach and then head to the intestines, and some of those materials are then absorbed into the bloodstream. Secretion, however, that's going to be when a solute moves from the bloodstream into the lumen of an organ. Last, we have a form of transportation across the cell, which is called transcytosis. And so using vehicles, the cell is able to move a substance into, across, and out of a cell. In this lecture, we learned all about transportation, how materials move in and out of a cell, across the cell, and we talked all about electrical chemical gradients, which are going to be very important once we go to other chapters and we talk about the function of certain organs and how these electrochemical gradients are going to dictate function. I know a lot of times this isn't the most appealing part of physiology, learning how solutes move across one side of a membrane to the other, but understanding how they do will help you in future chapters and in future lectures understand how certain cells are able to carry out functions. We talked about concentration gradients and electrochemical gradients in the metabolism chapter. We talked about the electron transport chain, and we talked about how hydrogens and the use of that concentration gradient was used in order for us to generate ATP. So there's going to be a lot of examples in the future where we use this molecular science, where we use the movement of ions and chemicals across one side of the cell to the other in order to explain what's going on in our cells, in our organs, and in our bodies. Thank you guys very much for tuning into this lecture today. I will see you in the next video.